Bioremediation of oil spills with microbes, not chemistry.
We are here today at the site of a very recent oil spill that has affected the giant goose population of Pope Park here in Hartford, Connecticut.
In the last 30 years, we have seen the devastating effects of many oil spills. From the Exxon Valdez of 1989 to the BP Deepwater Horizon spill of 2010, many people, animals, and environments have suffered significantly due to these disasters. We will explore the different techniques of how microbial biodegradation is used as a bioremediation method, and how the various microbes are able to break down and utilize the many components of oil. We will also look at the different techniques used to identify microbes in a specific environment, and how these techniques are used to help scientists determine which method should be used to help further clean up the contamination.
I want to be the very best at discovering the world, to explore the wilderness with my pen and my notepad. I will travel across the earth, looking at rocks and trees, to discover vast microbes and the power that's inside. Good afternoon, and welcome to another great episode of Magnificent Microbes and Belligerent Bacteria. I'm your host, Gil Nye, and I'm your other host, Steve Hurwitz. On today's episode, we're going to be discussing the presence of bioremediation and how it's increased over the past 10 years and used as a method of help cleaning up oil spills.
I'm Handy Pandy, and I'm from the DSL Oil Spill Research Association. Today I'm here to talk to you about the 1989 Exxon Valdez oil spill and the drastic effects that it had on the native people and native animals in the area. The 1989 Exxon Valdez oil spill had hugely negative impacts on people, especially the natives of Prince William Sound in Alaska. The categories mostly affected by the oil spill include contamination of foods, altered practices, treatment and cleanup, legal issues, restoration, commercial fishing, recreation, tourism, cleanup jobs, and the combined effects on the economy. The impacts on animals were even more devastating. We were lucky to get poor Diego out of the oil spill alive.
The oil spill not only impacted humans, but animals and habitats around Prince Edward Island as well, to a much larger and more direct extent. Recovery efforts are still happening for the animals today, 24 years later. The immediate death toll preceding the Exxon Valdez oil spill included about 100,000 animals, about 250,000 seabirds, 2,800 sea otters, 12 river otters, 300 harbor seals, 250 bald eagles, 22 orcas, countless fish, and trillions of small organisms like plankton.
Bioremediation is coming to the forefront of the cleanup process for oil spills because it is much more natural than the mechanical methods that are usually used. It uses much less heavy and expensive machinery and is safe and more delicate on the environments. Bioremediation looks as if it might be one of the most promising methods for beach cleanup because it causes much less damage and harm to the environments than traditional mechanical methods. It provides a much faster alternative to allowing nature to take its course because it simply speeds up the natural processes without having to introduce external methods.
Although it seems like bioremediation is the be-all end-all cure for the cleaning up of oil spills, there's still a lot of work that has to go into determining exactly which method of bioremediation should be used. There are companies out there that are dedicated to determining what species of microbes are in these environments where an oil spill is present. We're now going to hear from Catherine Microbia, who is the co-president of Mystery Microbes, a company dedicated to finding out what microbes are in these environments and how and exactly which methods are best to clean up these oil spills.
Thanks, Steve. I'm so excited to be here today to talk to you all about identification of bacteria in different oil spill environments. These microbes are so cool, and over the past few years, we've developed some great techniques for identification. It is interesting to study the bacteria in places like oil spill sites because bacteria are less diverse but more rare because the bacteria must be able to survive such toxic environments. It is difficult to identify these bacteria using cultural techniques because they may not be able to grow in vitro or outside of their natural environments. About 90 to 99% of the bacteria cannot be identified using techniques like this. Therefore, yet another form of identification is needed to help sort out the different species present in an oil spill environment.
There are two main molecular techniques microbiologists use. I will first explain the use of studying biomarkers such as phospholipid fatty acids. This is the way to identify specific gram-positive bacteria that are able to survive because phospholipid fatty acids degrade so quickly when an organism is no longer alive. Based on the unique fatty acid makeup, one is able to distinguish bacterial species. Let's take a closer look at a slide of some stained cells. The second main way to study these microbes is by studying the nucleic acid-based fingerprint of the 16S rRNA gene. This gene is highly conserved, as we saw with Carl Woese's research, and is conserved and found in all bacteria, but has small polymorphic differences based on specific species. We use a technique called denaturing gradient gel electrophoresis, or DGGE, to see and study these polymorphic differences. Let's take a closer look at DGGE techniques. After collecting samples from a particular site, the sample is prepared for PCR amplification. The 16S region is of interest in this case. Once the 16S region has been amplified through PCR, the double-stranded DNA fragments of the same length but different base composition can be separated in a gel. The gel used for DGGE contains a gradient of denaturing chemicals. These chemicals are urea and formamide. The DNA fragments migrate in the gel until they are partially denatured. The denaturing will occur at different places in the 16S sequence based on the polymorphic differences in the species. The number of fragments in the DGGE gel indicates the diversity in the microbial community. The DNA fragments in the electrophoresis gel can be sequenced. For this, the fragments are cut out of the gel, re-amplified, purified from primers and nucleotides, and then sequenced. This is an example of a completed DGGE gel.
Now that we've figured out how to isolate these microbes, we need to figure out what in the oil they actually like so much. We're here at the pond now where there's recently been a very bad oil spill, and we're going to collect a sample of the oil-ridden water so that we can take it back to the lab and test and see what compounds are actually present in this oil.
The composition of petroleum can be divided into four main categories. The first, known as saturates, are straight-chained alkanes that are most abundant and easily degraded by microbes. Branched alkanes are more resistant to degradation, and as the branch number increases, so does the resistance. Cycloalkanes, which are also within this category, are those with ring-like structures and are degraded at very slow rates or not at all. The second category are known as the aromatic hydrocarbons and are compounds containing one or more benzene or substituted benzene rings and evaporate easily, but are toxic to marine life and are relatively easy to degrade. The third category are the asphaltenes, which contain compounds that are either not biodegradable or are degraded very slowly. The material restricts the availability of oxygen and other nutrients, which make it hard for microorganisms to degrade. And lastly, resins, which are petroleum compounds that contain nitrogen, sulfur, and or oxygen.
I am joined now by my colleague, Christine Oceana. Dr. Oceana, it's a pleasure having you. Thanks for having me, Gil. Today, myself and Dr. Oceana are going to be analyzing the processes by which microbes break down oil. There are two types of processes. I will be discussing aerobic degradation, and Dr. Oceana will be discussing anaerobic remediation. Now, I'd like to refocus your attention over here. You see two types of mechanisms by which microbes are able to aerobically degrade contaminants in their environments. One that I'm going to be discussing, as I said, is aerobic, and as you guys know from previous episodes, that means that the final electron acceptor is oxygen. Additionally, microbes that degrade contaminants by aerobic respiration use enzymes that are specific to oxygen. These enzymes include oxygenases and dehydrogenases.
They are able to metabolize these organic compounds into final products such as carbon dioxide, water, and in some cases, small amounts of hydrogen gas. But how exactly does this happen?
The first step of this metabolic process is the use of a mono or dioxygenase to catalyze the addition of two oxygen molecules to the contaminant's benzene ring. The monooxygenase adds oxygens to the benzene ring in two separate steps, forming an intermediate arene oxide. Eventually, the monooxygenase forms the dihydrodyl, just as the dioxygenase does. However, as you can tell from looking at the dioxygenase reaction, there is no arene oxidized intermediate, as there was as formed by the monooxygenase reaction. Once this step is complete, the microbes utilize a dihydrogenase, an enzyme that is responsible for transferring hydrides to an electron acceptor, in this case, NAD+, which ultimately converts the dihydrodyl to a catechol. The microbes catalyzes ring fission and can then add a variety of substituents, either directly to the aromatic ring or to one of the created side chains. Ultimately, this results in the transformation of the original contaminant into substances that do not harm the environment. Through such transformations, electron donors become reduced and thus harvest energy for the microbes' metabolic processes. You might be wondering, how does oxygen really affect this process? In areas of high hydrocarbon contamination, levels of oxygen are often lowered, if not depleted. As a result, the microbes cannot produce ATP and therefore will not metabolize the necessary contaminants that are typically used as a source of energy. However, in a process called enhanced aerobic biodegradation, oxygen is added to the soil or groundwater in order to increase the vitality of indigenous microorganisms performing biodegradation. Therefore, organisms are not being added, but rather a necessary source of oxygen is added that encourages microbial growth and vitality.
Turn it over to Dr. Oceana and she can explain the processes of anaerobic remediation. Dr. Oceana? Apart from aerobic respiration, microbes are also able to perform anaerobic respiration, which, as you know, is when oxygen is not present. Let's take a closer look.
In anaerobic conditions, microbes metabolize oils and organic pollutants in the absence of oxygen. There are a few different ways that the microbes accomplish this. Generally speaking, the microbe will replace oxygen as the electron acceptor with nitrate, sulfate, carbon dioxide, oxidized metals, or even organic compounds. Today, we will discuss fumarate addition. Fumarate addition is when the methyl group of toluene is added to the double bond in fumarate to form benzyl succinate. The enzyme benzyl succinate synthase catalyzes this reaction. Succinyl-CoA is then added to form benzyl succinate-CoA. Acetyl-CoA is then added to form benzoyl-CoA. Finally, a ring cleavage occurs to form acetate. Then, through beta oxidation, the microbes can either produce carbon dioxide, form energy, make biomass, or make metabolite. Through this mechanism, microbes are able to break down oil or other organic compounds to make fuel in the absence of oxygen.
So, Steve, now that we've determined that microbes actually do break down the oil, I'm really curious, what do companies do to utilize these microbes to their advantage? That's a great question, Gil. I've actually brought a book with me today so that we can kind of go over some of the methods together. If you want to take a closer look, it really goes through them nicely. That's great.
There are two ways in which bioremediation by microbes is done. The first is fertilization, which is the method in which nutrients such as nitrogen and phosphorus are added to a spill site to stimulate growth and reproduction of the naturally occurring, biodegrading microorganisms. One of the limitations to natural biodegradation is the lack of nutrients that are seen naturally occurring in the environments and are required by these microbes to successfully degrade oil. Fertilization is thus the process by which these necessary nutrients are added to an environment. There are two types of fertilization, organic and inorganic. The first is inorganic, which happens when commercial fertilizers are introduced into an environment to accelerate the overall rate of biodegradation. The success of inorganic fertilization is marked by the changes in the composition of the oil over time and the overall respiration rate of the microbes. It has been shown that regular additions of fertilizer are much more effective than a single addition, but the rates at which the additions are needed has not yet been determined. The second is the use of organic fertilizers, which include Anupol EAP22 or other organic fertilizers derived from natural products like fish and meat. Anupol EAP22 was created and helps to prevent the formation of oil emulsions over time by reducing the viscosity of the oil. The additive supplies the natural microbes with the nutrients needed in order to successfully complete biodegradation processes. Other organic fertilizers made from fish and meat have more recently been identified and include up to 60% animal proteins with a nitrogen phosphate ratio that is known to be required by the naturally occurring microbes. Overall, organic fertilizers have been shown to stimulate growth and biodegradation by microbes much better in controlled studies than the inorganic fertilizers. The second main method, which is not seen nearly as often as fertilization, is known as seeding, or the process in which an environment is inoculated with a culture of naturally occurring microbes along with the nutrients needed for growth. There are a couple different ways in which seeding can occur. The first is done when a mix of indigenous and non-indigenous microbes that have been chosen based on the chemical makeup of the oil are introduced into an environment. It can also be done with a mix of naturally occurring microbes that have been cultured in lab and are introduced back into the environment. The main reason seeding is often used is to increase the overall population of microbes in that environment to hopefully increase the overall biodegradation rate. The advantage to seeding is that you are able to identify the specific composition of the oil and to choose microbes that will be best at breaking it down.
We're now going to take everything that we learned and compare two of the worst oil spills in history. The BP oil spill in the Gulf of 2010 and the Exxon Valdez spill in Alaska of 1989.
There are two big examples. One is the Exxon Valdez oil spill in Prince William Sound in Alaska in 1989. Approximately 41 million liters of Alaskan North Slope crude oil spilled into the Sound. This spill contaminated approximately 2,000 kilometers of shorelines between the Sound and the Gulf of Alaska. This spill successfully showed that bioremediation was a positive way to clean up surface oil spills. Addition of fertilizers was a key factor in the stimulation of microbe growth and therefore higher levels of oil degradation. Original concentrations of naturally occurring oil-degrading bacteria were between 1 to 10% of the microbial population. After addition, in late 1989, of fertilizer, populations increased to around 40%. In June of 1992, the USCG and state of Alaska declared the cleanup concluded. The other oil spill that we will be discussing today happened only a few years ago in 2010, so you may be more familiar with it. The BP Deepwater Horizon spill happened in the Gulf of Mexico. This was considered to be the largest accidental marine oil spill in history. About 780 million liters leaked. This was a deep sea spill, unlike the surface spill as in the Exxon Valdez. Dispersants were used in this spill. Dispersants disperse the oil at great depths and prevent large slicks from forming at the surface. This helps to prevent the oil from impacting the shoreline because biodegradation occurs on the water and oil interface. Dispersants increase the available surface area and in turn increase biodegradation.